Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to talk about eukaryotic cells. Recall that we talked about the three domains of life: bacteria, archaea, and eukarya. Eukaryotic cells are defined by the presence of membrane-bound organelles, the most notable being the nucleus. The word eukarya literally means true nucleus. This separates them apart from prokaryotic cells, which lack a nucleus and internal compartmentalization. Eukaryotic cells are found in all plants, animals, fungi, and protists, and they tend to be larger and more complex than prokaryotes. Some structural differences from prokaryotes that eukaryotes have is the nucleus, size and complexity, internal membranes, ribosome, and cell division. Let's start with the nucleus. The nucleus is where eukaryotes store DNA inside a nuclear envelope. In prokaryotes, DNA floats freely in the cytoplasm in a region called the nucleoid. Size and complexity. Eukaryotes are typically 10 to 100 picometers in size, while prokaryotes are typically 1 to 5 picometers. Eukaryotes also contain many membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, etc. Eukaryotic membranes have phospholipid bilayers. This helps compartmentalize processes and allows for specialization and greater efficiency in functions like protein synthesis and energy production. Ribosomes. Eukaryotic ribosomes are ATS, which means they're comprised of a 60S and 40S subunit. Prokaryotic ribosomes are 70S and they're comprised of a 30S and a 50S subunit. And finally, cell division. Eukaryotes divide by mitosis or meiosis, and prokaryotes divide by binary fission. Now let's talk about why compartmentalization is important. Eukaryotic cells separate different processes into specialized compartments, which are called organelles. This increases efficiency by allowing conflicting reactions to occur simultaneously, and it maintains optimal local environments, like acidic lysosomes and ATP-rich mitochondria. This also allows for complex regulation of pathways such as protein trafficking through the ER and Golgi. Think of it like a kitchen. Different tasks happen in different zones: chopping on the counter, cooking on the stove, washing the sink. If it all happens in one bowl, you would have a mess and wouldn't be able to do all of it. The same goes for cells. Here's a quick overview of the organelles of the eukarya. So it has a nucleus. This contains and protects DNA, does transcription, and does partial assembly of ribosomes. It has two membranes. We have the mitochondria. This produces ATP via the Krebs cycle and oxidative phosphorylation. This also has two membranes. Then you have ribosomes. These synthesize proteins. These have no membranes. You have the rough endoplasmic reticulum, which is the location of synthesis and modification of secretory, membrane-bound, and organelle proteins. Then you have the smooth endoplasmic reticulum. This functions to detoxify and break down glycogen in the liver and to synthesize steroid in gonads. And both endoplasmic reticulums have one membrane. Then you have the Golgi apparatus. This also has one membrane and it is used in modifying and sorting proteins and some synthesis. Finally, you have lysosomes and peroxisomes. Lysosomes contain acid hydrolases that digest various substances. Peroxisomes metabolize lipids and toxins using hydrogen peroxide, and they both have one membrane. That's it for today. Next time we'll talk about the nucleus.